I'm Miriam Heynckes and I have nothing to declare. My project is called Temporal Deviant Detection in Human Auditory Cortex using High-Field fMRI, and we are interested in whether layer-dependent responses in the primary auditory cortex are involved during behaviorally relevant detection in a complex auditory task. In this study, participants listened to narrowband sound sequences at two different carrier frequencies, here depicted in red and in blue, referring to low and high frequency respectively. We asked our participants to detect a temporally shifted target that was embedded in a quintet of tones, and these quintets would repeat at a 2 Hz rate. In a previous study, we have shown that periodicity is relevant to this detection. We know that detection is improved when the sequences are periodic compared to aperiodic. And what we are interested in now is whether the detection of this temporally shifted target is supported by a layer-dependent activation. The primary auditory cortex contains a map of frequency preference, which is called tonotopy, where neighboring neurons share a similar frequency preference. And we use this tonotopic mapping to understand whether regions that code for certain frequencies show a response across layers for behavioral detection of this temporally shifted tone. And we hypothesize that feedback, which is preferentially processed in superficial and in deep layers, would upregulate activity upon target detection and would do so in a frequency-specific way. And we do not expect to see this effect for the middle layers where the bottom-up input arrives. For this study, we scanned 8 subjects at 7T using a sparse auditory design with a TR of 3.5 seconds, where the sounds were presented between the acquisitions. After every sound, the participant was cued to respond whether they detected a target or not. We acquired a slab of 0.8 submillimeter data centered on Hesch's gyrus bilaterally where the auditory cortex is located. In addition, we acquired a tonotopic localizer centered on the same location, as well as whole brain anatomical data using the MP2 RAGE sequence. We performed classical preprocessing of the fMRI and obtained a gray matter white matter segmentation with manual corrections around Hesch's gyrus to delineate accurate equivolume depth layers. For the analysis then, we estimated the response to single trials using a GLM. To estimate the best frequency of every voxel, we employed tonotopic mapping on the main experiment and on the localizer. For tonotopic mapping, we select voxels that respond to the presentation of sounds and then z-score those responses spatially in order to remove biased responses to low sounds. Here I'm showing you an example from one participant. We defined primary auditory cortex based on the frequency gradient obtained in the tonotopy, as described in Morel et al.'s paper. And within this region, we then select voxels preferring low and high sounds. Here I'm showing you the low preferring voxels. On the right then, you can see the data from the main experiment at the level of betas before spatial z-scoring in the top row and after spatial z-scoring in the bottom row. And we can see that this region maintains the frequency selectivity between the localizer and the main experiment. Our pipeline then allows us to extract layer profiles to our experimental conditions in frequency selective regions. Here I'm plotting the average responses to the low and high sounds in the low and high preferring regions across cortical depth on the x-axis. The shading is the variability across participants. And we can see the typical gradient echo EPI signature with a response increase towards the surface. The relevance of extracting single trials in the analysis then comes in when we analyze the response to detected and undetected trials separately. Here, I'm showing preliminary data from one hemisphere and one participant, again for the same regions for the separate sounds. Magenta refers to detected trials, blue refers to undetected trials, and green refers to trials where there was no target. And we can see that the magenta line shows a larger increase towards superficial layers. And this appears to be in line with our hypothesis that detection of behaviorally relevant sounds would be accompanied by an increase in the superficial layers, although very preliminary data. To summarize, we validated here that the tonotopic localization allows us to define stable high frequency and low frequency preferring regions, and we can investigate responses to behaviorally relevant sounds in a frequency specific manner. Moreover, our preliminary results show a typical gradient echo signature across depth and a larger increase towards superficial layers in detector trials. Our next steps would then be to repeat this analysis, depth analysis for every participant, and to explore the effect in regions outside the primary auditory cortex. And with this, I would like to thank my supervisors and my colleagues, as well as the funding agency. Thank you for your attention.